Hello, welcome to this evening's Science Unwrapped. We're very happy to have you in this spring semester as we celebrate grad student research. I'm Professor Brynja Kohler, Chair of the Science Unwrapped Committee and also Associate Dean in the College of Arts and Sciences. And on behalf of the college, we're really excited to have you here this evening. So thanks for your time. Thanks for coming. A graduate student, for those of you who might not know, is someone who's already finished a college degree or a bachelor's degree, and they're currently working towards a master's or doctoral degree at a university. Sometimes we call a doctoral degree a PhD. This is a very challenging pursuit, and it involves lots of hard work and perseverance. Utah State is very grateful and proud of our graduate students who are vital contributors to our teaching and research missions. During this series, we have been highlighting several grad students along with their faculty mentors who are conducting fascinating studies. Did you know that next week is actually National Engineers Week, and USU's College of Engineering is going to be celebrating with activities each day, including a free community night on Thursday, February 26th at 6 p.m. in the Taggart Student Center. So our featured speakers this evening are engineers, and I imagine there's others in the audience who are engineers. Would all the engineers please stand? All right, let's thank our engineers. Engineers make the world a better place. All right, now it's my pleasure to introduce our speakers. We're very excited to have graduate student researcher Akib Mahir and his faculty mentor, Dr. Liyuan Joanna from the department. Akib is from the Department of Civil and Environmental Engineering and the Department of Biological Engineering here at USU. Akib is a doctoral student and serves as a graduate research assistant at the Water Research Laboratory. His research focuses on microplastics and microbial biofilms and how these communities form on plastic surfaces and how they affect wastewater and aquatic environments. Prior to joining USU, Akib worked in the Bangladeshi banking sector as an environmental risk manager. Outside the lab and classroom, Akib enjoys spending time with his family and playing chess, and he also has a passion for pigeons of all kinds and would happily talk to you about that hobby. Dr. Joanna Howe is an assistant professor of biological and environmental engineering at Utah State University's Water Research Laboratory. Her research addresses challenges with microplastics and bioplastics using molecular tools and data-driven approaches to explore how these materials shape microbial ecology in water and soils. She is deeply committed to undergraduate and graduate student research mentoring and actively involves students in hands-on laboratory and field studies. Please welcome Akib and Dr. Joanna Howe.
Thank you. Okay, welcome everyone. It's my honor to do the Science Unwrapped event talk tonight. So hopefully I can give you a exciting and also enjoyable talk to start your weekend. Okay, so my name is Liyuan Hao. You can also call me Joanna. That is easier for you probably. So I have my PhD candidate students, Akib and Mahir here, and we are going to present you some, bring you to a world, explore some little tiny things, but with a big impact, which is microplastic. But instead of just talking about plastic pollution, I want to let you think about something you probably never think about before. What happened? Microbes or germs jump onto those microplastic and use it as a taxi. Okay, So before we dive into that word, I want to show you where is my lab is. So each lab is actually housed or located as the Utah Water Research Laboratory. Have you been to the first dam? Raise your hand. Yes. Okay, so if you're passing the first dam, you will see the Utah Water Research Laboratory there. That is actually the oldest water center in the United States and the largest one, and one of the largest one and one of the oldest one. So that means we study water. We study pollutions, we study microbes, and how those affect or how to protect our human and environmental health. So if you are interested, we actually have free tour as well at the World Lab, so you can scan the QR code to if you're interested, and then you can explore more in the Utah Water Research Laboratory. And then on that picture, you can see me, Joanna, and also you can see Akib. So we are from different country. We have a different background, but we share one thing. We are curious about little tiny thing that can change the world. Okay, so emerging contaminants, that sounds a really technical term, right? Sounds really complex, but that is newly emerging pollutants that we are still trying to figure out or study, okay? So that includes microplastic, nanoplastic. But we are more than that. We care about the microbes. We care about how microbes interact with those emerging contaminants. Okay, quick challenge here. Raise your hands up if you have used some plastic products today. All of you, right? Okay, keep raising your hands if you drink water bottle. Okay, if you use the to-go box, which is made polystyrene. Some of you use phone cases made by PVC. Okay, most of you. And now mention those like laundry detergent containers or peel container, they are all made by plastic. And I want to remind you, some of our clothes is made by nylon. So when you wash those clothes, those nanofibers or microfibers will break off from your clothes, went to your washing machine, and go to the wastewater treatment pipeline. And also even tea bag. Some of the tea bag is made by plastic, so they will have plastic fibers in there. Okay, so plastic is woven in our daily routine. Okay, so normally, often we just didn't realize that. Okay, so plastic is so cheap. Okay, it's so durable, it's so convenient. So we love it. We love it so much. But the thing is, those little tiny particles, plastic particles coming from those things, they don't disappear easily. They last and last. I want to show you some data. We produce about 400 million metric tons per year globally of plastic. Only a small fraction, 9%, ended up goes to recycle. Most of them go to landfills. Okay, some of you don't know landfills. We just bury them under the earth. And then still, most of those plastics, they actually leach from those landfills and goes to the environment, like the water, soil, and eventually in the animals, and eventually in our human beings. Okay, so every year we have 30 to 50 million metric tons of plastic waste are mismanaged, and those larger pieces of plastic during sunlight, during waves, physical forces, and germs, microbes, they slowly break them into smaller pieces, and then those tiny pieces will go into water, soil, and the food chain. Now I want you to see those pictures. Okay, some of the images actually from Akib's hometown. So he has seen this in real life. Many countries which has limited resources for wastewater, plastic waste recycling has these plastic pollutions. And then Akib, instead of just upset about this plastic pollutions in his country, he decided to study about it and how to deal with that. So Akib, would you like to share with us why this become more personal for you?
Yeah, sure. Thank you, Dr. Howe. So, hi everyone. I'm Akib, and I'm from Dhaka, Bangladesh. And the picture that you're seeing here, right here, is from where I'm from. It's from Dhaka, Bangladesh. On the left picture, it is from a publicly used road, and the right picture is a water body. And the thing that we can see here is just waste, right? So this is not only in Bangladesh, this is a problem in all of the developing countries, because in the US, the waste management system is very structured, there's infrastructure, there are landfills, but in developing countries where the population density is very high and the land area is very low, we don't have that much area for landfills and stuff. So waste management is a big challenge for us. And as you can see in the pictures, most of those, what do you see? Plastic bags, right? Just like plastic bags right here. And why do we have them so much? Because they're so easy to use, they're so durable, they're so handy, and they're so cheap. And that's why we make them so much. Then after using them, we dump them, and it happens, it goes into the environment like this. And it doesn't stay like this, right? These things, they break down into smaller particles, and they end up into different water bodies, into the lakes, ponds, and eventually to the oceans. And then it does not stay as a problem for the developing countries like Bangladesh. Then it becomes a global problem. It's polluting the oceans, and it's causing harm to environments overall. And as an environmental enthusiast, as a conscious person, I wanted to study how things are being affected like this by plastics and how we can manage them, or what are the factors affecting them. So that inspired me to pursue my higher studies, grad studies here at the USU, and fortunately at Dr. Howe's lab, who's also working with microplastics and microbes, and that has been the inspiration more for me coming here at the US.
Thank you, Akib. So now you see, this is not just a scientific problem. It's a real communities, real rivers, and real consequences. Okay, when you hear about microplastic, how small do you think it is? Like a rock? Like a grain of rice? Okay, yes, it's a really small. So, microplastic, okay, is a plastic particles between 0.1 millimeter to 5 millimeters. That sounds really technical, so let's make it really simple for you. 1 millimeter is about size of a gram of sand, and the 5 millimeter is about the size of pencil erasers. Okay, so now imagine something even smaller, like dust. Okay, so microplastic is just smaller like that. Okay, and they are hard to see, hard to be removed from water. Okay, it's a pen. And even some of the plastic particles, they become nano-sized. They are nanoplastic. You cannot use your naked eyes to see it. You have to use special equipment to detect it. But today, we're going only to tell about microplastic, because microplastic themselves is already a challenging part. Okay, now we know microplastic are tiny, but where are they actually coming from? There are two major sources. One is a primary microplastic. Another one is secondary microplastic. So primary microplastic, they are made small on purpose. Okay, so ladies, I want you to think about, have you seen any smaller microplastic fragments or beads in your cosmetics, in your facial cleaning products? Those are the microplastic, okay? And then the secondary microplastic is actually coming from our laundry machine, when we washing our nylon clothes, or those larger particles, they are underwent those like sunlight waves, physical forces, or even chewing up by those microbes or germs. They become even smaller and smaller. One thing I want you to remember, they don't disappear, okay? They get smaller, they get smaller. Here is something many people don't realize, that not all plastic are the same. They vary in shapes. I want to show you. They have fragments, they have fibers, films, pellets. More than that, there are various of shapes that you can imagine. Another thing is they vary in materials. So we have polyethylene there, right? We have PVC there, we have PET there, we have polystyrene there. We have all kinds of different polymer types. Okay, why I mention that? Because shapes can affect how the microplastic travel. I want you to imagine one scenario. Can you imagine the threshold fiber when they drop in the water? Will that behave similar as the solid beads? Probably not, right? And also, the different recipe of the polymers like polyethylene, polypropylene, polystyrene, they have different surface chemistry, okay? And their density is different. Some of the plastic, they are denser. If you drop off any plastic type bottles in the water, some of them they sink, okay? If they are denser than water, they sink. If they're lighter than water, they float. So you know, some of the microplastic, they always float on the surface of water. Some of them, they sink, okay? Now we talk about where microplastic goes. I always got one questions from my friends or families knowing I'm doing microplastic. They always ask, can microplastic enter our body? The answer is yes. Yes, okay. So when you drinking water, okay, when you eat some seafood, they have those microplastics. Or even you breathe, there's some microplastic in there, okay, it enter our body. But the scientists are still studying the long-term health effects of those microplastics. So some studies showing that they can bother our lungs, and also they can carry some germ or pathogens that can make us sick. Okay, so that makes this scenario more complex because microplastic, they never travel alone. Okay, raise your hands if you hear about the word germ before. Okay, do you think germs are bad? Okay, I see some nod hand and shake hand. Microbes, some what we call germ. Some of them are good, some of them are bad. Okay, they are more than that. Okay, so they are in water, soil, and inside of us. Okay, you cannot see them because they are so small, but they are constantly working for our Earth. So a human hair is about 70 micrometer wide. A bacterium is about 1 micrometer. So that you can imagine about like 70 bacteria, they can line up is equal to the width of the one hair, okay? So they're tiny. So when microplastic enter the water and the microbes saw them, what do you think? They treat them as a trash? No, it's treated as a surface, okay? So they will attach on the surface, they will grow, and then they will build communities, okay? They're microbial communities. And those communities called biofilm. So it's a tiny microbial city, okay? It's crowded with microbes. Have you ever been fuzzy, feel fuzzy stuff on your teeth in the morning when you brush your teeth? Yes, okay, I saw a nodding hand. Have you slept on the green algae on the stairs? Yes, okay. Have you seen slime on the rocks in the water? Yes, yes. They are slimy layer. They are biofilm. Okay, they are biofilm. So those microbes attached to the surface, they will produce a sticky surface, biofilm. Okay, so those biofilm, when they form on the microplastic, that make microplastic a city, a living city, and that is what we study in our lab. So once biofilm formed, they will form layer by layers of the microbes, and those microbes will attach on the surface, and they will produce something is called glue. And then those glue has a terminology, the name is called extracellular polymeric substance. Don't worry, don't need to remember that at all. But I want you to see what's in there. There are proteins. There are some carbohydrates, which just sugars, and then DNAs. Those compounds are denser than water, so they may causing the biofilm also denser, and also it attach on the surface of microplastic, so it will affect the density or the falling behaviors of the microplastics. And then another thing is those microplastics can carry those microbes around the world. Okay, now imagine this. Imagine this. You are a microbe. Okay, you are a microbe. There's suddenly a new plastic surface show up, just like taxi. What will you do? You will jump on it. Take a ride, right? Why not? So then this microbe ecosystem living on the plastic, we call it plastosphere. So if some of you are really interested in searching for plastosphere, okay? And then, so think microplastic just like a taxi, and the microbes hook on that. So they can ride through the rivers and through the lakes to the oceans. But here's a twist. The passengers can actually change the taxi, okay? Change the taxi. Some of the passengers are good. We mentioned the microbes. Some of them are bad. Some of they're good. They can carry on breaking up those large pieces into smaller pieces and choosing them up as a food. And some of them, they have a denser biofilm density, so they will causing those microplastics to settle. So which means naturally just remove them in our natural system. But some of them, they are pathogens. They're bad. They're dangerous microbes. Okay, they can kill. They can causing us to be sick and carried by those microplastics. And some of them, they're antibiotic-resistant bacteria, which means even you ask doctor for prescriptions for antibiotics to try to cure you, but it cannot because it's antibiotic-resistant. So taxis, microplastic can carry all of them. Okay, but they have preference. Sometimes they prefer this type, sometimes they prefer the other type, but we don't know. Is that interesting? So the key message I want you guys to remember is microbes can change the microplastic taxi moves, change their density, falling speed, how they transport. Another thing is microplastics can also select or have a preferential of which passengers I want to carry because their surface chemistry is so different. So that means this plastosphere is not static. It's a dynamic word, okay? It's a dynamic. It's constantly changing, which is really fascinating because if we know that, we know the biofilm-covered microplastic is an ecological moving system. That means they will determine whether those plastic float on the surface of surface water or sink in the sediment, right? And also, they will influence how the microplastic travel. And also, we can see which bad passengers is on that, giving us some, environmental health risk predictions, right? But what we don't know is, we don't know a lot. We don't know exactly which microbes make microplastic heavier. We don't know how size and shape influence biofilm growth. We don't know how the wastewater microbes may have a lot of bad passengers on it, change the microplastic movement. And we cannot yet predict how biofilms alter the microplastics behaviors. So to answer those big questions, okay, so we actually team up with experts from multidisciplinary. For example, we have Dr. Tim Burke from the Mechanical Aerosol Engineering Department. He is really good at monitoring the falling speed of microplastic in turbulent water. So it's not still water. Turbulent water is like a turbulent a lot. It's actually the real scenario in the ocean, in the surface water. And we can also provide our data, like, for example, the biofilm density, the microplastic density, and then send it to our experts, Dr. Yanqin Su, who is an AI modeling expert, which can use our data, generate that modeling, and so that we can systematically quantify and model how the microplastic carry with those potential bad passengers and move in the environment. And then our role, okay, my role and Akib's role in this team is trying to answer those three questions. The first question, which type of microbes make microplastic heavier or lighter? The second question, how microplastic size and shape type affect floating and sinking? Third, how wastewater microbes change microplastic movement? Okay, starting from this point, I'm going to hand the microphone to Akib. Let him to give you what our research plan, and we have some really exciting preliminary data, and we want to show you.
Thank you, Dr. Hao. So our research involves 3 questions, but instead of seeing them as questions, I would like to view them as are there any gamers here who play computer games? Oh, I see quite a lot of hands. So I'm a gamer too, and I like to see this from a gamer's perspective. So in games, we have missions and objectives that we have to fulfill, right? So this is the game we are playing, Microplastics, Biofilms, and we have missions, 3 missions or objectives that we want to see what's happening there, what's going on there, and we want to do some lab experiments to answer those questions. So the first mission or objective that we have is that which type of microbes makes the microplastic heavier or lighter. So in the environment, there are like various microbes. The system is not controlled. Many environmental factors are coming into play. So to make it simpler, to make them easy to understand, we want to have a lab-controlled environment, right? So we can control the factors and how they behave. So for doing that, for answering the first objective, we have selected 2 pure species of microorganisms. Like there are various microorganisms out there, millions and millions. We know some of them, don't know many of them. So to understand how biofilms are formed on microplastic, we select 2 pure species and microplastic form from one type of polymer, the PET. The water bottle you see here, this will break down into smaller PET particles into the environment. So we will collect PET particles, procure them or make them in the lab, and let them 2 types of microbes grow biofilms on them. So one of them is a bacteria. The one you can see on the left is the Pseudomonas aeruginosa, and another is a cyanobacteria or an algae, the Microcystis. So these are like commonly found in lakes, water, soils, and the bacteria one is kind of pathogenic, and it might cause infections if you're exposed to it. And the bacteria one forms a denser biofilms. The biofilms they'll form will be a bit heavier or a bit denser compared to the cyanobacteria one. So if you have been to the Bear Lake, there's some, during the summer, there's some algal blooms in there, right? Those yucky green things in the water that we don't like to touch. So those are like algal blooms, and these microcystis, they are sometimes responsible for those algal blooms in there. And we want them in the lab, and we want to grow the biofilms on microplastics and compare which one is making lighter and which one is making denser biofilms and how they're affecting the microplastics. So this is our first mission, the first objective, and then we will go into the second one. So in the first experiment, we were using 2 types of microbes and 1 type of plastic. But in the second objective or second mission, we are going to use different sizes and shapes of plastics, but keep the type of microbes as one. Then we could understand how the different sizes and shapes are affecting the biofilm growth, and how the biofilm is affecting different types of plastics, different shapes of plastic. So there's an AI-generated image right here. You can see like various shapes of microplastics, right? There's some circular ones, the spherical ones, beads, and there's like threads as well. So we will be producing different shapes of microbes. The table indicates what we will be using, PETs and PEs of different sizes and shapes, and we will see how microbes are interacting with the different sizes. So the second image is also AI generated, and we also hope to see that due to the difference in shapes and size and how they're formed, or the polymer type, the biofilm formation on them will be different, and they will affect the microplastic differently. So this is our second objective that we want to see, and on the third objective, we want to see the real world scenario, because in the first 2, the conditions were strictly lab controlled. We have like 1 type of plastic, 2 type of plastics, 2 microbes. But in the real world, the microbial community is diverse, right? There are various microbes interacting in the water bodies, and they're interacting with the plastics in different ways. So we want to see what the real scenario is like and how it affects the microplastics and forms biofilms in the environment. So this image right here, can anyone tell me what this is an image of? It's a wastewater treatment plant. We have one in Logan. And the second image, this is an image of the effluent system. So dirty water comes into the wastewater treatment plants. They do some chemistry processing, and voila, the water is cleaned. compared to how they came in, and then they're released into the environment. And interestingly, this water also has a host of microbes in them. So for our third objective, we will collect wastewater samples from this effluent system and culture microbes. We grow the microbes in the lab that are present in the wastewater system and let them grow biofilms on the microplastics. And then we'll compare how the pure species and how the microbial community and the environment, how they're differently forming the biofilms and how they're affecting the microplastics differently. So that is our third objective. So it seems pretty easy, right? 3 objectives of a computer game. I think we'll be done very shortly. But unfortunately, no, the process is very tough. And I want to see how or what we do in the lab. So we are talking about growing biofilms on microplastics. Where we will do it, how we will do it, we'll see in this slide. So where's my pointer? Yeah. So this is what we call a drip flow bioreactor. So what this does is this is the place where we put the microplastics and the microbes in and where they interact with each other and grow the biofilms. And this right here are the microplastics that we have been talking about. I have some right here in this little petri dish. If anyone wants to see, we can pass them over and you can see what we work with in the lab. These are like microplastics. And we put them in these chambers, right? We have 6 chambers per bioreactor, and we put them, and this right here is the microbial culture. We collected the wastewater, we took some of them and let the microbes in there grow, and we have all those microbes growing in here, and there's this media. You can consider the media as a bed for the microbes and the microplastics to sleep on, where they will interact in these chambers, and in these chambers they will form biofilms, and we will give them some food. So how it works, the total system looks like this. We have these jars right here, which are called influent. We have this pump, the bioreactor right here, and the discharge. So what we do is for the microbes to grow, they will definitely need food, right? They will need food to grow and interact with the microplastics. So we are providing the food. Where's my pointer? So yeah, those jars right there, we are providing the food through them, and we using that pump, we are pumping those water drops, which has nutrients for the microbes to grow, microbes to feed on, and we are pouring them into each of those chambers, and the microbes are using those as a source of nutrient or as a source of food, and they're growing and developing biofilms on the microplastics. And after we have grown them, this is what the microplastics and the biofilms look like. In the bottom picture, look at those. There are some sticky, gooey substances. Are you able to see those? Those sticky substances? Those are what the biofilms are, right? So in the left two side, on the right side, they're like a greenish, brownish type of stuff on the microplastics. So those are the microbes forming biofilms on them. And on the middle one, the one that says no microbes, see how clear the plastics are? There are no biofilms growing on them. And if we look at them under a microscope, on the right side, you can see, so there's an image of a microplastic without biofilms, and on the bottom picture, this is what the microplastics looks like with biofilms. So you can see those sketches and you can see those sketches like pencil drawn in something. And if we look at them like closer, or if we want to use a stronger microscope, this is what we have. So this right here is a scanning electron microscope. This is quite a powerful microscope that we have at USU, and with its help, we can look at the microplastics and the microbes and biofilms all together, and this is what it looks like. So this is a plastic particle, 1.5 millimeter in diameter. So these are very small. I have passed some, if you could see, those are very small particles. Under a scanning electron microscope, this looks like this big surface on 100 times magnification. And if we magnify further, if we go into 5,000 times magnification, we can see all the microbes that we have been talking about right here. See, those shapes, each of those are bacterial or microbial cell that has grown on the microplastic. And if we look even closer, 10,000 magnification, this is what it looks like. Those are the individual cells. And interestingly, in 10,000 magnification, we can see something different as well. We can see some webs or sticky things around the microbial cells. So we wanted to look closer and see all of these waves are the things that we call the biofilms, right? The sticky substances that we have in the morning sticking to our tooth, we brush it off. So those are the biofilms, and it looks like this under microscope. So after our experiments, we want to see, visualize, and this is what they look like under a microscope. And then we have the microbes, we have the microplastics, we have the forming biofilms, and now what do we want to do with them? So we had three major questions, and to answer them, we want to look at what the biofilms are made of and how it changes the plastics. So Dr. Howe already mentioned that the biofilms are made of proteins, carbohydrates, DNA, and cells, and we can actually see them, right, under a microscope. Yeah, they're made of cells, there are proteins and carbohydrates in there, and it is definitely changing the microplastic. When we normally look at a microplastic with or without biofilm, we do not understand the difference. We don't know if anything's there. But under a microscope, all those things are there, right? Thousands and thousands and millions of cells, and they are definitely affecting the microplastic in some way or another. They can change the surface roughness, because if I put this table is bare, it's normal, right? If I put a layer on it, then the surface roughness changes. And similarly, the microbes are changing the surface roughness of the microplastics. Then they are changing or making the microplastics lighter or heavier, or they're changing the density. And they are by affecting the density, they are also affecting how fast or how slow they fall in water. So we have some preliminary data from our experiments. So the first images show a clean PET particle, which is from a plastic bottle, and the one with biofilm formation, the yellowish things are the biofilms. And on the right, we have a graph. So the blue bars represent protein content, and as we said, biofilms are made of protein, right? So higher protein content means more biofilm growth, and lesser protein content means less biofilm growth. So the blue bars represent the protein content, and the red line over there represents the falling speed. So on the first bar, you can see that higher protein content has a lower falling speed for the PET particles. And as the protein decreases, the falling speed increases. And a clean PET, where there's no biofilm, it has the highest falling speed. So this is quite interesting. Due to the formation of biofilm, the falling speed of the microplastic is what? Decreasing, right? So we did more experiments. We wanted to test, okay, this is happening for PET. How does it change for polystyrene or PHA? So for those, we did those experiments. We took those microplastics, we have grown biofilms on them, and then we measured their density, and we observed how they fall in the water. So we'll look at this video. If you can see those arrows right there, indicates where the microplastics are falling from so that you can understand cleanly. So look at this video. So what we see is the microplastic on the left is blown away by the turbulence or the water flow, but on the right, it goes through the flow and settles on the bottom. So the left one is a clean polystyrene particle, and the right one is a polystyrene with biofilm. And in our experiments, we have seen that the density of the polystyrene particle increases after the formation of biofilm. So we have measured the density, and it increases, and from our experiments in falling water, we can see the difference, because the lighter particle, which is lesser density, is blown away, but the one with a higher density, it settles into the bottom. Now, we will look at another video, and here there will be a race going on, and you have to look at those to see which one comes first and which one comes second. Start. Oh, the left one is reaching the bottom faster. Oh, the left one is the winner. So on the left side, we have the microplastic without biofilm, and on the right side, we have microplastic with biofilm. So what's the difference in this case? Compared to the last video, we had polystyrene particle in there, but this one is a PHA. So this particle or this plastic has a higher density compared to the polystyrene, and due to the formation of biofilm, their density eventually decreased, which led to the decrease in falling speed. So from these two videos, we can assume that microplastics and biofilms, they interact with each other differently. Each type of microbe, each type of plastic, their shape and their size affect their falling speed and how they form biofilms. And this has great implications in wastewater system, in the environment, or also in the engineering processes. So from all of our experiments that we did and the objectives that we have, we can see that these tiny things, these tiny microbes and the microplastics, they can do big things. They have severe implications in the real world. And through the advancement of science, we can see those, we can measure those, right? Through the microscope, we can see all those tiny, tiny cells that wouldn't have been possible to see. So science is a wonder, and we are doing some wonderful things with it. And I can see many young people here. And if you guys are interested, you can also dive into this. You can learn science and try to figure out what's happening out there. There are microplastics, nanoplastics, microbes. There's so many things to study about. And you can dive into this field. You can try to explore and solve a global problem one way or another. So thank you, everyone.
Thank you, and we are really appreciative for your time here tonight. And then our project actually is major funded by the NSF Environmental Engineering Program, and then we got some funding from somewhere else as well. And then thank you for your attention. Thank you. And for someone got the little container with microplastic, please return to me because I don't want to have any plastic pollution.
Okay, thank you, everyone. We'll get started with questions. If you have a question, raise your hand, and our ambassador, Arts and Science Ambassador Jake, will come to you with the microphone.
Go to mommy, go to mommy.
I don't know how to phrase this question, but I guess there was a time when there weren't plastics. So I don't know exactly how they make plastic. Like, what is the original product they start with? Some petroleum byproduct? And whatever product they start with to make plastic, can they ever break it down to that original product? Like, when you say some of the microbes break down microplastics, they break it down to what? I mean, to a particular molecule or anyway?
Yeah, sure. So microplastics, when they break down, so the breakdown process involves eating the microplastic or having them as a source of food. So when we're eating food, like a chicken, so we eat chicken, we take the energy from them, and we have the byproducts or the waste products. And similarly, the biodegradation process that we talk about. So this bottle, a PET. So PET is degraded by one type of microbe, and they will consume the PET, use it as a source of energy, and they will release some byproducts, terephthalic acid and stuff. Not the actual carbon and hydrogen. These are polymers like carbon and hydrogen products, right? So when they're like degrading, so they have different byproducts. Not the actual original one, but eventually every organic matter can be. What's the word? It breaks down eventually to the nutrients, but the microbes that are engaging with them directly do not break them to the original products that they came from. They have some byproducts.
I want to repeat the question first. You say you are not sure where those microplastics coming from, right? So we said two sources. One is from they made it are small on purpose, like facial cleaning products, cosmetics, or they're breaking forcefully from other factors. For example, during the wind, during the sunlight, the UV will have oxidation, so they will break those polymer chain and break those chain part. So they may become smaller particles, or in the end, they probably will become monomer or oligomers. And then if monomers or oligomers can be eaten as a food sources by microbes, they will become carbon dioxide eventually. Yeah, eventually.
So polymers are carbon and what else?
Yeah, carbon hydrogen bonds and some other.
Most of them, they are carbon bonds or carbon ester bond or carbon oxygen bond. Yeah. Yes.
What sample sizes were you using in most of your experiments, and what did you classify as an observation? Was it a microbe? Was it a piece of plastic? Was it a group of plastics?
Okay, so for our experiments, I'll just go onto the slide and so that you will be able to understand easier. So these are the microbes, right? Microplastics, sorry. So we have like 3 of these bioreactors, 6 chambers each, and after putting the microplastics, growing the biofilm, we do different type of experiments. So we talk about protein content, carbohydrate content, DNA, density. So each of those are a separate experiment. We have to, we need 30 microplastic particle for doing DNA, 25 for protein, and 30 for carbohydrate, and for density we use 10, 15 as much as we need. So in each of those chambers we have around 90 to 100 microplastic particles. We put them by counting one each and put them. So multiply that by 18 and we get the number of microplastics that we see. And the microbe, we put the microbes in there. We do not count the number of cells. We use optical density as a measure, and we have a specific optical density for our experiments, OD 0.33 that we use, and that is for each of those chambers. But it will vary based on what type of microbe we are using, or is this a microbial culture from wastewater. So yeah, that is how we are doing the experiments.
Great question. Really scientific. Any other questions?
Thank you for your very interesting presentation. You're talking a lot about microbes that are naturally encountering microplastics in the environment. I'm wondering also about the possibility of genetically modified microbes that could be engineered to eat plastic. Obviously, those aren't going to be natural, but we've heard about the promise of this for a few decades, and I'm wondering where that is. If you could speak to that.
Yeah, that's a really good question. So you are asking if there are some microbes, we can engineer them to ask them just chewing down those microplastics, right? Yes. So for the PET, which is the one-time use bottles, there's already microbes that has been reported to do the work and has a special enzyme, which is called PETnase. And then Nature journal already published one engineered E. coli species has that enzyme to do the work. So it's not a dream, it's already come true. And then they are optimize that enzyme, which the downstream enzyme METnase, to make it even chewing the PET faster. But the problem is we have so many type of plastic polymer type. Polystyrene, PVC, polypropylene, polyethylene. So far, polypropylene, polyethylene, polystyrene has been reported some microbes can do that work, but not all the plastic types. I think a lot of the plastic types, they're stubborn. They probably will be on the earth for a long time. That's the microbe can do nothing with it. But we potentially, yes, we can do the engineering species to do that. Yes.
Sorry. Okay, so he asked about creating them. Is there some evidence that it's happening organically, that the proportions of bacteria in the atmosphere who can do something with these plastics is just increasing because of evolution and changes in the environment? Do we even have data on that?
Fantastic questions. I don't have that answer. I don't think so far we can evaluation or make. But I want to point out one thing is not bacteria. There are many microbes in the world, like fungi. Some fungi, they're so special. I want you to think about in the forest. You see fungi grow on where? On the wood, right? On the lignin cellulose. And those lignin cellulose polymers, some of them, they have similar structure as polymers, which saw here, which is petri-based polymers. And those extracellular produced enzyme from those fungi, they actually cannot differentiate, oh, this is the polymer type of lignin. That is the polymer type from one of the plastics. So they do both work. So sometimes it works, but I don't have the data for like naturally how they evolved to become available to do this type of work. But that is a really good question. Other questions?
Hi, really quick, just thank you for that presentation. I was wondering what your method was for determining the density of the microplastic after it had developed the film on it, because typically, I assume that once the film had grown on the plastic, it would have changed the shape of the surface, and typically on like a macro scale, you could use a liquid displacement test, but I assume that wouldn't be feasible at such a small scale, so I was just wondering how you determined the overall density.
Yeah, that's a great question, and we have struggled for quite some time for the accurate method where we can accurately see the small change in density, right? So due to formation of biofilm, the density change wouldn't be that large. So as you say, the displacement gets kind of tough. So we did some literature review, and we found a great method where we used a buoyancy. So the microplastic, suppose polystyrene. The polystyrene has a density roughly equal to that of water, normal water. So we took the microplastic particles of polystyrene into methanol, which has lower density. So those, due to the polystyrene particles being higher in density, those sank in the ethanol. And then we added a saturated sodium chloride solution and dropwise until they were in kind of this state, like they are not sinking or they're not floating. So we can assume that the density of the microplastic particles and the liquid is similar. So then we measured the density of the liquid, which we are assuming to be the density of the microplastics with and without biofilms. And in both of those cases, we have seen the difference. The polystyrene particles behave differently, the density rating came out differently, and and the density number was different. So this is how we did it. Yeah, thanks.
Does the microbiome on the film that grows on the microplastics accelerate in colonies? So where the microplastics are more abundant, like in landfills, does the breakdown process multiply and like the process from beginning grow and expand throughout, or is it like better in small environments where it's more controlled, like in a laboratory?
So if you're asking about like biofilm formation on the microplastic, then it is more on water surfaces, mostly in water bodies. The microbes are there, but in soil, microbes might grow, but the thing is, the degradation process that we talk about, microbial degradation of the plastic, this is kind of a slow process, and the amount of plastic that we have in the environment is so large that they will break down, and some of this will be broken down into smaller monomers, but eventually the bigger plastics break down into smaller ones, and then they go into degradation. But the whole process is very large, or very time consuming and lengthy. So microbial degradation can alone never be a method where we can remove all the plastics from the environment. So as you asked, if it's happening in soil, in water, in mostly the water bodies, the biofilm will grow more and affect how they travel. But yeah, there will be microplastics and there will be microbial colonies on the surfaces. But the studies are quite rare, but most of them have been done in water bodies compared to the soils.
Can I add something to that? I just want to let you guys know the original source of this project. So the plastic in the environment right now is breaking apart. We can do nothing about it unless you go in person and collect it, right? The only places that you can think about harvest or return those microplastic is actually wastewater treatment plants, because that is where wastewater goes, having those microplastics, and then we treat it. We have already infrastructure to do that. That's the reason we are more curious about in the waterways, because in the wastewater we already have clarifier, which is a settling tank. And then if we understand their falling speed and density, probably we can just use those equipped, right, built system to solve this microplastic problem. So that's stopping those microplastics enter the environment from wastewater, from human beings.
All right, let's thank our speakers once again. This was a fascinating evening, so thank you very much.
Thank you,
Joanna.
everyone, for coming.